Okay, hi everyone. Good morning, and it's a real pleasure to have Pontus Skoglund here. And just briefly, we've been knowing each other for a long time. Pontus did his PhD on population genetics in Uppsala with Matthias Jakobsson and Ancient DNA, and then he spent several years in the Harvard team with David Reich, and then got his own as a group leader, a position at the Francis Crick Institute, where he still is with the USC grant. And he's been doing a lot of fantastic work on many different parts of the world, the Americas, the Pacific, and also in the last few years, more on dogs and wolves. And he's gonna give the lecture on fine-scale genetic history.
Yes, thank you very much, Ron, and the entire department HEAS for the invitation to talk today. Seems like a very exciting environment. It's great to be here in Vienna. And yet today, I would like to talk about fine-scale genetic history, how we can understand ancestry with ancient DNA in higher resolution, why we would like to understand fine-scale genetic history, and what some of the examples of things that we have been recently studying and discovering has been. And today's talk is really based largely on this paper that we've made available as a preprint, based on a new method that we call TwigStats to understand high-resolution ancestry. And this is a project led in my lab by Leo Speidel, who has been a postdoc with me jointly with UCL over the past few years, and also a collaboration with archaeologists and other geneticists to try to understand archaeological and historical interpretations of what we're seeing. And this is really a new method to try to obtain higher resolution of genetic history. And what I'll also try to convince you of is when we apply this to some real data, the data that we can find where there's the best available whole genomes at the moment, we think, in Northern Europe. I'm going to try to convince you that we can see genetic evidence in ancient DNA of expansions of Germanic speakers in the first half of the first millennium CE. So that's CE, the Common Era, or AD, Anno Domini, the same thing. So about 2,000 years ago, it's the start of the first millennium. We can see those expansions southwards in the eastern and western directions. And later on in the first millennium CE, I'll try to convince you that we can see evidence of an expansion into Scandinavia as well. That hasn't really been apparent in ancient DNA data as of yet. But I'd like to first motivate a little bit why we want to understand ancestry and how we can do it. And it's to make the point that ancestry is very useful for understanding history. If we imagine that we knew the two parents of every person who's lived in, say, the past 10,000 years, you can imagine that that would be a really useful resource for understanding historical processes, of course, also beyond the historical record into where we only have archaeology. And the good thing about genetics is that through our genomes, our chromosomes, as depicted here, they can trace ancestry throughout this family tree. But if we had really, really rich information about the family tree, we would have to really have tried to piece apart broader patterns. On a per individual level, it's a very kind of messy and complicated view. A very dynamic genetic history is what ancient DNA has uncovered in essentially all examples that have been studied so far. But it's something really worth getting into. And the advantage that our genomes provide is that while there's a single unifying family tree of humanity, right, a pedigree, it's a single one, everyone's two biological parents, our genomes, different segments, different genes, can trace different paths through this unifying family tree. And so that means that even a single genome provides more information than sort of just one trace through the family tree. It provides multiple different ones. And so we need to have whole genome information. And also this fact that different segments trace different paths through ancestry, sort of evolutionary speaking, provides also an evolutionary variance. And that means that when we talk about statistical uncertainty, it's often about covering as much of the genome as possible. When we want to understand our statistical uncertainty, we will want to resample across the genome. It's a small technical point. And I think also conceptually, what is ancestry? At least the way I like to think about ancestry is not just about what, like that a person is of some particular ancestry or of multiple particular ancestries, but it will always be a question also about when. Ancestry is different in different temporal perspectives. Perhaps 60,000 years ago, for example, perhaps 2% of my ancestry would find itself in Neanderthals. And the other 98% might find itself in Homo sapiens in Africa or its vicinity. 6,000 years ago, it will be a different picture. Many of you will have heard about the numerous ancient DNA studies about hunter-gatherers, pastoralists, and early farmers in this time frame. We understand that with some detail, for example, in Europe. But more recently, in the past few millennia, we know less. And it's really important to have this kind of temporal perspectives. It's about different time slices, and it's going to be something that changes ancestry flows through time in dynamic ways. And on the left here is a summary from a few years ago about some of the really continental scale evidence of dynamic genomic history that's been uncovered so far by ancient DNA. But there's also a lot of regional scale history. And in this panel from the right here by a project called the People of the British Isles Project, which was really about modern DNA, is an example of hypothesized historical processes that might be traceable in genetic history, but is really pushing the limit of where we have resolution with ancient DNA. And I'll get back to that. Because one way of thinking about how much statistical power do we have to understand genetic history is that it also requires us to be able to distinguish between ancestries, right? So you could imagine that there's some really dynamic genetic history. Perhaps you could imagine a regional replacement of groups. Some groups come in, completely replace another group. But if they are very genetically similar, close related, not so differentiated, we might not be able to tell by looking at the genetics. And one way to think about, to sort of quantify this, is the proportion of genetic variation that is between groups. And groups is something that's always loosely defined. But here's, for example, a few examples of Stone Age groups that you might have heard about in the context of ancient DNA studies, particularly then sort of hunter-gatherer, early farmer, and steppe groups. And you can see that the proportion of genetic variation between these groups is between 5 and 10%. And that's actually quite a lot. It's not on the level of what you might see between different animal species in nature, but it's quite a lot, and it's plenty of statistical power. And that's why you might see also ancient DNA studies that are really focused on more recent time periods, still kind of using these ancestries as tracer dice of later historical processes. Because if we go back to early medieval Europe, or sort of the first millennium CE that I mentioned, if we take a few loose groups here, for example, comparing Scandinavia to Britain or Scandinavia to France, it's an order of magnitude lower genetic variation. Only 0.5% or so of the genetic variation is between groups. And that's really a statistical challenge. It makes it difficult to trace genetic history, and it's kind of an impetus for developing stronger, high-resolution ancestry methods. And there are some approaches that do provide more power to detect patterns in ancient DNA and modern DNA, as in this case. The same study, People of the British Isles Project on modern DNA, was able to find really fine-scale patterns in people today in Britain by focusing on longer segments shared between individuals, finding, for example, really fine-scale patterns here in the southwest of Britain. Also, I was involved in a project in the same year where we found this fascinating signal for shared ancestry between people in the Amazon today and people in Oceania and Southeast Asia. And on the right-hand side, you can see two different analysis QQ plots where we expect sort of a random distribution to fall on this dashed line here. And in the top here, you can see some of the groups that show the largest evidence of shared ancestry with the Amazon. And when we did it in a conventional way on so-called SNPs, SNPs, single nucleotide variants, you can think of it as a sort of single mutation where an A changes to a T or a G changes to an A. We had reasonable evidence, but we were kind of worried about this. It's a very fascinating signal. We wanted to boost it, work to try to find many different ways to boost this signal. And one way was, again, this kind of chromopainter segment sharing approach, where we observed a much stronger signal, kind of a 6 sigma result for this particular comparison. And so that's segment sharing, again, showing evidence for a strong genetic link. But we couldn't really quantify, what was the proportion, what was the genetic history, what was the proportion ancestry in the Amazon related to something different outside of the Americas. We couldn't do that with these kind of segment sharing approaches. And another recent development, for example, in this paper by Harald Ringbauer et al., there's also ways of looking at even longer segments over millions of bases in our genomes shared between individuals. For example, in this case, uncovering evidence of sort of sharing or relatedness on an individual level between individuals associated with different cultures on the steppe and in Europe about 4,000 to 6,000 years ago. And these are very powerful to detect pairs of relatives, pairs of shared ancestry between individuals, to some extent between groups. But again, we can't really quantify it in this view, sort of how 60,000 years ago, 2% of my ancestry is Neanderthal. It's harder to do that with these types of methods. And so the opportunity that we looked at in Leo's project was to use these new genealogy methods, where you don't just look at shared segments, you also get temporal resolution of how segments are related to each other. And so then this is a tree reconstructed from one part of the genome, and each tip on the bottom here is a different human chromosome. And then you can reconstruct how they trace the path through the shared family tree together, and each blue point is a mutation that allows you to kind of temporally resolve and date these different shared ancestry events. And on the average in the human species, the time to the most recent common ancestor of everyone will be over 500,000 years. But what if we would focus on recent shared ancestry? And now, instead of looking at just long segments, we actually have temporal resolution. We know when we cut off the time at which we look, perhaps we can gain unbiased ancestry models based on that. That was kind of the rationale we were following. And so one way to look at this is that we were trying to find a solution for understanding population level ancestry models, how much ancestry and at what point in time, but not just in the realm where conventional F-statistics models have been so successful, where genetic differentiation is on the higher side, but also when it's on the lower side. And also trying to fill a gap where segment sharing methods are powerful for detecting individual relatedness, but really trying to build it into the realm of population level ancestry models. And here's the first example of applying this method. First, it's based on using SNPs, single mutations, to try to reconstruct some genetic differentiation. And this is a kind of clustering method displayed in 2 different dimensions, on the x-axis and the y-axis, trying to differentiate between different groups in Neolithic Europe, early farmers, from different regions. And you can say that it does a pretty good job at separating some of the major patterns here. But when we apply this cutoff, we only look at ancestry shared ancestry more recent than a certain point, we can see new patterns. For example, a fascinating pattern that was also found in the original paper by Laura Cassidy et al., where people in Ireland, buried in particular passage grave, show more close relatedness than people in Ireland and Britain found in other contexts. Really sort of partitioning and finding some patterns in the data that's not apparent if you just apply SNP-based methods. So that's quite encouraging. But as I mentioned before, we really want to get into model-based ancestry, because these types of clustering methods or principal component methods, while they are very useful for finding things in a sort of qualitative way, they have limitations. And this illustration is an example of that, where I have simulated a genetic history where we have one ancient DNA sample that's clearly more closely related to these red present-day individuals. And the ancient individual is also colored in red, more closely related to the red present-day people than, for example, the blue individual simulated here or the green here. And if we do a principal component analysis of this data, you can see it kind of mirrors that. This individual seems to be more closely related to these red individuals here. But if we change the scenario and introduce a bottleneck, a crash in the number of individuals here, that will sort of push away the present-day red individuals. And actually, it might seem even that the ancient individual is slightly closer to the other group. So methods like clustering and principal component analysis are sensitive to what we call genetic drift, stochastic events that speed up whenever population size is smaller, or what we call effective population size is smaller. That means higher degree of genetic drift, faster stochastic and random evolution that tends to push things away. And really an amazing solution to this has been F-statistic-based methods pioneered by David Reich and Nick Patterson about 15 years ago now, that have evolved in this approach called QPADM, which I've tried to illustrate here, where we can apply very generalized ancestry models, ideally having more ancient individuals in a group of a set of individuals or populations indicated here in these different letters. And then we can have a study individual and ask the question, well, how does it relate to these individuals? We can ask if it is consistent with having ancestry from just one of them, perhaps two of them, or perhaps three of them. And it's also possible to test if the model is consistent with the data, because if one of the other individuals or groups that were not modeled as sources has a closer relationship to the study individual, that will often result in a rejection of the model. And so the best case scenario is that we try all of these different pairs or combinations of test sources and find that only one model fits the data. All the others can be rejected. It's a really powerful tool. And if I was studying ancient DNA and could only use one tool, this might very well be the one I would use. And so to test tweak stats, Leo did this simulation, really along the lines of using this type of approach, QPDM-like approach, or in this case, an F4 ratio, which is a more specific case, where we simulate data under a particular admixture proportion, in this case 20%, and we estimate ancestry based on also simulated data under the same model. And on the right here, you can see on the y-axis the inferred admixture proportion, and the black dot here is the estimated ancestry proportion using conventional SNPs, again, single nucleotide markers, single mutations, but you can see that the error bar is really absolutely massive. So there's too much statistical uncertainty in this case. We can't actually say if this individual is maybe 100% of this ancestry, or 100% ancestry from population 1 or 0% ancestry from population 1. We have no statistical certainty about that, not enough statistical power. But if we apply the temporal time restriction, cut time off at a particular point in time, we get these error bars. Really a radical improvement with twig stats, and these 3 different estimates are doing it slightly different way. One of them is using the true genealogies, one of them is using inferred genealogies, using this tool called Relate that Leo developed when he was a PhD student with Simon Myers. And the final one is using mutations on the tree that are dated, but that's sort of technical details. And you can also see that it's unbiased, as far as we can see in these simulations. It's also been unbiased when we apply it to cases such as early Europe, quantifying hunter-gatherers, steppe pastoralist, and farmer proportions. And also, we tried 2 other approaches, one based on only taking rare variants below a certain frequency in the simulation, and one based on taking shared segments in the simulation. And you can see that both of them are biased. They are actually estimate higher proportions than the true value. Really an effect of this bottleneck type effect that I showed you in the principal component analysis as well. The advantage with this, what we call the twig stats approach of time restriction, is that it's temporally resolved, and so we avoid a lot of these biases. And we think that the reason for rare variants also being biased is that they are not necessarily young. So here, Leo has quantified the ages of particular mutations, and you can see even if they have a minor allele frequency here on the x-axis that is really quite low, some of them can be really quite old still, up to 10,000 generations easily, and they're not temporally resolved. They can still introduce some of these biases we see. Okay, so I've talked about having a time restriction, cutting off time at some particular point. What's that time? How does that make sense? And we find that intuitively you can think of it as the optimal cutoff point is just before the divergence of the sources. Many people will have heard about admixture from Neanderthals. So that's the example chosen here, where some people have some proportion of their ancestry from Neanderthals, which means that if you trace their ancestry back in time, in some segments of the genome, that segment will trace its ancestry into Neanderthals, finding itself in Neanderthals at some point in time, just as I said before. And if it finds a common ancestor with a Neanderthal chromosome, that's important information, right? We can see it shares ancestry with Neanderthals. However, some other segments might also find themselves having shared ancestry with Neanderthals, but actually in the common ancestor, not due to this admixture. And they are not informative of this, do we have 2% or do we have 10% Neanderthal ancestry? And so the ideal cutoff point is just before, going forward in time, the divergence of the sources. That's really the shared ancestry that's relevant when we're trying to quantify these ancestry proportions. Of course, who is the sources is something that might change depending on our temporal perspective if we want to understand ancestry 60,000 years ago or 6,000 years ago. And just to show here about the optimal cutoff point, Leo did some simulations and developed theory that shows why it's actually not exactly at the divergence time of the sources, but just before that, since some of the shared ancestry, some of the coalescences that happen in the ancestral population are informative about dating events that happened in the sources themselves. As a second application, in addition to Neolithic Europe, we were interested in deeper human evolution history. Can we learn things about that as well? And that is this question about Neanderthal admixture, which already by the original Neanderthal draft genome paper, it was highlighted that one of the explanations for the sort of shared ancestry between people outside of Africa and Neanderthals is, of course, admixture in the ancestral population, so scenario 3 here. But another possible explanation in theory for those patterns could also have been if there was deep structure, where Neanderthals, for example, had a long period of contact with, let's say, Homo sapiens in Northern Africa or Eastern Africa, that then brought some of that shared ancestry with them outside. And that's been recently brought up again as something that these authors thought were consistent with the data they saw. And so we decided to do a simulation of this, try to see if cutting off time at different places could reveal something about this. Can we show that whether shared ancestry is recent or further back in time? And in a Neanderthal admixture simulation, we see that, for example, finding 2% ancestry is consistent regardless of when we put the cutoff time. So, for example, in generations here, from 100,000 generations to 8,000 generations. You can see all of these lines are fairly straight. But in this alternative deep structure simulation, where there's no second contact or admixture between Neanderthals and Homo sapiens 60,000 years ago, both the true trees and the inferred trees have a behavior where we don't see any evidence. We don't infer this 2% ancestry if we put the cutoff time very recently, some 10,000 years ago, 10,000 generations ago. Okay, so then we can go into real data. If we do the same with real empirical human genomes, what do we see? Which one of these two do they match? And you can see that we see the same straight line as in the Neanderthal admixture simulation, really providing quite strong evidence for a punctual admixture event. And I think even without these types of analysis, there's evidence of long segment sharing with Neanderthals that I think would show the same point. So one thing about this genealogy-based method is that it requires whole genomes. But actually, most ancient DNA, if you've heard about there being over 10,000 individuals, ancient people with genome-wide data, the majority, absolute majority even of that is sort of genome-wide capture of single nucleotide polymorphisms, about 1.2 million of them. There's much fewer whole genomes. This is sort of a fake curve I put in. I don't know exactly the numbers, but I think it's about 2,000 whole genomes at this point. And so it's only a fraction of that, but they are really the only way we can apply this method, we think, in a robust way, because if you only have a sort of selection of SNPs, you will have some subset of these mutations on the tree. So the temporal resolution might actually be skewed because of ascertainment bias. We need whole genomes, we think, to do this genealogy-based approach. And to me, what we see as really quite successful applications of this method is really an incentive, I think, to really focus on whole genomes going forward. And so then I will turn to the main application that we were interested in this applying this method to, which was Europe in the first millennium CE, because of previous amazing papers that have sequenced whole genomes. Should be over 1,000 shotgun whole genomes available from this, and mostly from the northern half of Europe, which is also the reason why we focus on it. And we turn to a few different questions using this data, which is, can we understand demographic processes during Germanic expansions, which I'll get back to? Can we search for evidence of unknown expansions beyond the Roman frontiers, beyond those documented by Roman historians? And can we obtain more individual level ancestry portraits in the Viking Age, which previous studies have already shown were really, really diverse, really high evidence of mobility, people of different ancestries in different places. And another interesting point about this is that with higher resolution methods, we can also move away from modeling the ancestry of, for example, early medieval groups or individuals using present-day groups. If we, for example, would model ancient individuals using the high sort of tens of thousands of genomes available from the present day, there's a danger there that actually what we're capturing is processes that happened after those individuals lived. Right? If I say that an ancient individual is closely related to some particular country, how do I know that that's because of things in their history and not things in history since they lived? Right? And so we really try to focus on modeling early medieval groups or individuals using sources that are dated to the Iron Age, sort of preceding periods or approximately contemporary periods. And I think as we build an even bigger ancient genomic record, we'll be able to do this even more confidently. And so we started collecting this data, and first define some different source groups. And the broad patterns are illustrated in this kind of two-dimensional model of genetic affinities. And you can kind of see in the upper left corner here individuals in the Iron Age that were on the Scandinavian peninsula. On the right-hand side, more Eastern European ancestries, and on the bottom, Southern European ancestries, and kind of in the middle, Central European ancestries. And in this inset here, you can see sort of within Northern European clustering, if we do it, on the right here, if we do it just with point mutations, single SNPs, and on the left, if we do it with genealogies, you can see there's already some improved resolution in finding different groups. And so in the following figures, here are the colors that will be used for showing particular ancestries. So this kind of pink color for ancestry related to the Scandinavian early Iron Age, red for Britain, this yellow ancestries for Central Europe, which is really quite interesting place to understand more about in the future as well. And then also, which you will see less frequently, purple, green, and blue colors for other regions. And so to kind of try to walk you through what we see here, or some of the interesting patterns, at least we thought, was that similarly to what has been seen by some previous studies, in Poland in the early Iron Age, individuals associated with the Wilbar culture, which has indeed been associated with the historical Goths, which were Eastern Germanic speakers, we see a high degree of ancestry related to the Scandinavian early Iron Age. And as I'll show you in the next slide, that's really quite an ancestry change compared to the Bronze Age. And again, later in medieval periods, there's a quite drastic ancestry change. In medieval individuals available at the moment, we see little or no ancestry from the early Iron Age Wilbar associated individuals, likely due to expansions of Slavic speakers into the region. And we also see this expansion of Germanic speakers into present-day Slovakia, which represents a change from earlier Latin-associated individuals. Particularly, it's fascinating Zohor individual, which I'll have on the next slide as well, associated with early Germanic speakers, which was sequenced by a paper this year. And what's fascinating is that if a bit later in time in the migration period, there's a mixture of these sources. So what we tend to see is a kind of a punctual expansion of these, perhaps of these Germanic groups, followed by mixture and sort of absorption with other groups, perhaps coming into the area or previously local groups. And that's something I think we've seen in the ancient DNA record in a lot of different time periods, and a lot of different regions, sort of an initial expansion that's quite punctual, followed by more permeable contacts between groups. In Central Europe, we really see mostly continuity in many ways with earlier Iron Age periods, but we do see some evidence of change as well. Perhaps we need to understand them with high-resolution data later. But one thing that's fascinating is a few individuals associated with early Germanic groups in present-day Bavaria, southern Germany, show a much greater evidence of ancestry related to the Scandinavian early Iron Age, again indicative of these expansions, but also again demonstrating mixture with this kind of yellow-colored ancestry from previous Iron Age groups. Similar things are seen in Longobards-associated individuals from Hungary and the Pannonian Basin that also show evidence associated with Scandinavian Iron Age indicative of these expansions. And another case people will be familiar with and which has also been studied before is Germanic expansions into Britain associated with the Anglo-Saxons, showing a very similar pattern as well. But fascinatingly, there's one individual from actually the previous periods in Britain, Roman periods, from this interesting gladiator cemetery previous studies also noted that it seemed like an outlier. And in our hands, it indeed seems like it has Scandinavian early Iron Age ancestry already in the 1st or 2nd century CE. But to go back to Poland, sort of like looking at it not with these ancestry-based models, but again, a more sort of model-free approach, a principal component-like approach. So we have time here from left to right. And you can see that at the end of the Bronze Age, or in middle to late Bronze Age in Poland, ancestry is very much more similar to general sort of Eastern European ancestry in this time period. And the early Iron Age wheelbark individuals represent a quite radical shift to that, which then changes again in the Middle Ages, as I mentioned previously. And in at least the data we have currently from the present day, which is a bit thin, seems to sort of have a relative continuity with the Middle Ages. And this fascinating Sohor individual, which was sequenced by this great study by Antonio et al., which Ron's group, was a collaboration with Ron's group as well, dates to the 1st to 2nd century AD. I think it's interesting because it's in Slovakia, so kind of at least in the data we have, the southernmost extension of this Eastern Germanic expansion. And indeed, archaeologists and the original paper noted that this was likely the grave, a grave associated with a Germanic settlement area, this female 50 to 60 years old, and really quite different from the ancestry that you might see in sort of in southeastern Europe around the time. In our hands, modeled as having 100% ancestry from the early Iron Age in Scandinavia. And you will also see that it has sort of this slightly lighter pink color with a diamond symbol, which in our hands is representative of a slightly northern associated type of Scandinavian ancestry. We could tell the difference between two broad groups of Scandinavian early Iron Age ancestry. We don't know the exact geographical distribution of them. With more individuals, that will be possible. But certainly, this one is sort of more distributed, probably to the north. And it turns out that if you look at, let's see, sorry. That if you look at the pink colors here, you can see that this diamond and light pink color is much more prevalent in the southeastern direction. Whereas even in the Longobard-associated individuals here, the Bavarian-associated individuals here, as well as a little bit less certain in Britain, it's the other sort of more southernly associated color. And this could be perhaps a first sort of evidence that of what linguists identify as sort of Eastern Germanic-speaking groups that expanded in this area, eventually reaching present-day Ukraine, and Western Germanic-speaking groups that expanded in this kind of more central direction and to the west as well, which is, to me at least, very fascinating that we can perhaps start to tell the difference of these really fine-scale patterns. And one more fascinating thing you'll see here that we're not entirely sure of at the moment, but it's quite compelling to us, is evidence of this kind of purple ancestry in these Central European or South Central European Germanic groups. And that ancestry is represented best in our data by Sarmatian-associated groups. So an Iranian-speaking group of people in the 1st millennium CE that were distributed in roughly these regions, which you can see is quite close to those Germanic-associated individuals we were studying. Something that hasn't really been shown, that perhaps the presence of these groups in the 1st millennium CE might have included quite a bit of contact with other local groups and be found as mixture in later individuals. And this group's also fascinating to us because in our ancient DNA lab in London, Marina Silva was studying a lot of data from Roman periods and Iron Age periods in Britain. And there was one individual in a principal component analysis that didn't fit with other individuals that are in this sort of red cloud of points here, but it's this yellow rectangle over here, really having ancestry most similar to what you find in regions in the Pontic-Caspian steppe or the Caucasus. And we really think that the best explanation for this is indeed in ancestry models as well. This individual is best modeled as having Sarmatian ancestry, and perhaps he represents an evidence of mobility across the Roman Empire to Britain. And what's quite specifically fascinating about this is that the individual dates to around the time of the Marcomannic Wars, where in 175 CE, Sarmatians were defeated by Rome at the end of these wars in this kind of area. And the emperor Marcus Aurelius deployed these defeated Sarmatians, 5,000 of them, to Britain to sort of fight the northern frontier here. It's something that's been mentioned historically, but not much has been, no individual of Sarmatian ancestry has been found so far. And so it's really a fascinating example of that type of mobility almost 2,000 years ago. Sorry, so here's a sort of a better picture of this kind of eastern and western directions we can see, and perhaps how they might relate to previous hypotheses on Eastern Germanic expansions in this direction that would later in the migration period also impact, of course, the areas of the former Roman Empire as well, where you can see sort of the two different shades of pink being differentially represented. It's something that's a little bit on the edge of our resolution at the moment, but I think it's a very promising sign when new data becomes available. And so then moving forward in the first millennium to the second half of it, where I mentioned in the beginning, we see evidence of an expansion into Scandinavia in approximately this time period. And here is particular bars showing the proportion of ancestry from early Iron Age Scandinavia in Viking Age individuals in Scandinavia. And you can see that indeed there's lots of continuity. For many individuals, we can see that they have ancestry that's best represented by the early Iron Age in the region. But there's also a lot of individuals that showed ancestry from Central Europe or continental Europe. We don't know exactly where it could be from. It could be from sort of northern Germany. It could also be more southern parts of France, South Central Europe as well. And you can see that it's really distributed in the southern parts of Scandinavia, Denmark, southern Sweden, the sort of Oslo fjord here as well. And it's something that's not seen in any individual that predates this period. So it looks to us as if there was some expansion event that hasn't really been understood before from the continent into Scandinavia. We see it quite clearly also in Denmark. You can see it with this model-free approach where Iron Age individuals are really in the upper left corner here with the Scandinavian early Iron Age, but the Viking Age individuals have a wide distribution reaching into Central Europe in a very clear way. And when we look at ancestry models, this looks a bit complicated, but on the top row, we use SNPs, and on the bottom row, we use twig stats. And you can see that for example, this group, this region in Denmark, if we use two ancestry sources, there are multiple different models that all fit the data. So we can't tell between them, all of these different sources represented by different colors. But if we apply the twig stats approach of time cutoff, we get this resolved to only one model fitting the data, which really aids our interpretation. And it's consistent with this influx of continental associated ancestry into Scandinavia. And when do we think this happened? Well, where do we not observe it? We do not observe it in individuals in Denmark that are dated to about sort of the 2nd or 3rd century CE. Also individuals from southern Sweden a little bit later. We do not see it in individuals from about 500 CE on Öland here, from this fascinating massacre site at Sandbyborg. I'll get back to Öland in a second. And we also don't see it in individuals associated with this ship burial sequenced by the Copenhagen group led by Eske Willerslev. Fascinating data from present-day Estonia here, but they are historically thought to have originated in central Sweden as well. So we don't see it there. It must have come to, well, at least not been sort of fully represented in these regions until these times. I mentioned Öland, which is this island in the Baltic Sea, which also offered a fantastic opportunity because there's a great study by Wilhelmsson and Price from 2017 that did stable isotopes from over 100 individuals, which greatly overlaps with data generated again by the Copenhagen group. Really great to have access to all of this data, which allowed us to ask the question, well, is these individuals that carried continental type ancestry, were they just sort of like newcomers into the region? Are they evidence of mobility, or were they locals? Did they have stable isotope signatures that were indicative of having grown up in this region due to kind of absorbing drinking water with the same mineral content as you would expect to find in this region? And so I'll try to walk you through this slide, which is an example of trying to merge ancestry data on a per individual level with stable isotope data. And so there was quite a degree of variation in ancestry in individuals from Öland, from 100% continental related ancestry to 0%. And so that's kind of the y-axis here and these dots. And then in the stable isotope data, you can see this histogram where there's kind of two modes, one kind of majority group here and one other group here. With different isotope values. And you can see that the majority group had stable isotope ratios, strontium stable isotope ratios around this range here, which is really consistent with the local sort of expectations from the local geology. Whereas the individuals over here had isotope ratios more consistent with coming from elsewhere, perhaps, for example, from northern Scandinavia or something like that. So then we thought, well, maybe these people with continental related ancestry are the people that have stable isotopes indicating that they are newcomers to the region. But actually, it was the opposite. So all individuals that have 100% continental ancestry had stable isotopes more consistent with the local geology. In fact, there's a really quite strong trend that locals tended to have higher degrees of continental ancestry. So this was not just a transient phenomenon. These were people who were somewhat established in these regions of southern Scandinavia, something interesting to understand with future studies. And here sort of just trying to make the point that with high resolution methods, we can get more individual level understanding, which I think will hopefully also be useful to archaeologists that want to understand local context. Here's two individuals from Öland. This individual had a high degree of Scandinavian early Iron Age associated ancestry, also Eastern European ancestry. Again, a very high stable isotope ratio, suggesting that he was not a local. And this individual had definitely evidence of continental, or in this case, actually was unresolved, could even have had Southern European ancestry, but had evidence of a sort of stable isotope ratio consistent with the local geology. And I wanted to show this one also because while even on Öland, there are individuals where we have a single model that we can resolve ancestry to. For some individuals, multiple models are consistent with the data. And I think that's really important to show the level of uncertainty we might sometimes have. And so this individual definitely did not have that much ancestry from the Scandinavian early Iron Age. And so it's an example of this phenomenon of expansion. If we look elsewhere in Viking Age Scandinavia, we can see evidence of mobility, individuals that are somewhat rarer, but evidence certainly of the mobility in this time period. Again, some of these patterns have definitely been noted by the previous excellent studies that have looked at this. For example, we see evidence from Iron Age Britain, which would have been sort of Celtic-speaking periods in Britain, all across Scandinavia, indeed overrepresented in the northern Norwegian coast. which could be consistent with sort of seafaring contacts. Some evidence of ancestry from Southern Europe, and certainly also evidence of ancestry related to Eastern European Iron Age, particularly in the eastern parts of Scandinavia. And it's exciting to me to be able to see these ancestry models on an individual basis, really something to really merge archaeological contexts with ancestry in the future. And one fascinating example is also beyond Scandinavia, looking at this people with significant evidence of ancestry related to Iron Age Britain. And here splitting it up into females and males, and each sort of rectangle Leo has plotted is one individual with evidence of such ancestry. And if the rectangle is white, there's individuals that do not have evidence of it. And we can see evidence of ancestry related to Iron Age Britain as far as Lake Ladoga in present-day Russia. And you can see it also elsewhere, particularly on Iceland. It's fascinating because you might have thought, based on some hypotheses, that most of the individuals with ancestry related to Britain would have been females. But in fact, that's not really what you see in the data. I think it's been noted by previous studies as well. Plenty of males had ancestry related to Iron Age Britain during the or at least later on in Iceland, perhaps during the colonization of Iceland as well. And so to summarize again, applying high-resolution ancestry methods, we think we're able to see now with genetic high-resolution methods evidence of Germanic speaking expansions in the first half of the millennium, of the first millennium CE. And probably a bit later, an expansion into Scandinavia that was not really documented by historical sources so far. Perhaps in the future, looking at historical sources in a new way might be able to find out more about these processes. So I'll just talk about one more thing. It's a bit more technical nature, because how can we now take these higher resolution methods forward to understand new things about genetic history? And one of the things is that we will need whole genomes, so-called shotgun data, as I mentioned before. But there's also an aspect of how to, sort of a technical aspect of how to prepare and extract ancient DNA. And one really great approach is to use this treatment that removes damage due to cytosine deamination that's really common in ancient DNA. And this type of damage is distributed towards the end. The treatment is very effective at removing it in most places, but actually, to go into the molecular biology for a second, UDG treatment cannot treat cytosines that have been methylated. And that's the case methylation for the majority of these variants in CpG context in the human genome. And so for the ancient DNA researchers that in the audience, here's the kind of classic figure of cytosine deamination. But this is actually on UDG or user-treated data. But if you stratify it out and look at CpG context variants, they have full damage. They're completely unrepaired by UDG treatment, and still have full damage, and that kind of error process that can be a bit tricky, I think, to apply. And if we think about applying high resolution methods, where we're now looking at really recent shared ancestry, that is also going to be rarer genetic variants. And then these kind of slight error profiles might make it more difficult to do that. In the empirical applications I've shown, we have removed all SNPs that could be subject to this. But if we really want to boost our statistical resolution in the future, it would be really nice to also do high resolution ancestry analysis with all variants in the genome, also including this. And one of the things that we can do then is, based on single-stranded DNA library preparations, which were developed by Matthias Meyer and others in Leipzig, which allow you to really stratify things out and only remove specifically these Ts, these thymines, looking at the strands, looking at their strand orientation to the reference genome, sort of like removing half of the data in an unbiased way, which we can talk about if there are technical questions. And that's something we've been doing a lot with the ancient DNA generated in our lab. Ancient DNA researchers might know about imputation, where we sort of try to reconstruct the genomes using panels available to us. And we find that in these two curves here, you can see the triangles are when we do sort of single-stranded DNA library aware genotyping and imputation. And we see much better accuracy in the genotype imputation when we do this. We also see that if we don't repair or we don't do this correction, we can actually draw the wrong conclusions based on ancestry. In this principal component analysis, if we don't correct for damage, ancient DNA damage, these dots are actually shifted away from the sort of affiliations of the individuals when we correct for the damage. So that's going to be, I think, something that might really improve the resolution of our data. And actually, the issue is that you might think this CPG context doesn't matter so much, but actually even on things like the 1.2 million SNPs, that's most of the ancient DNA data today, about 25% of them are transitions inside CPG context. That's only really correctable with single-stranded DNA libraries. UDG treatment will unfortunately not correct it. And so while we, as in the applications I've shown today, can do really well with just the transversion SNPs, about a third of human genetic variants, we can get really high resolution inference. We can get even more access to even more mutations with UDG treatment. We can get access to the most variation if we also include single-stranded DNA-aware approaches. And so I think that's really could be the way where we correct for this single-stranded DNA. We use single-stranded DNA library-based methods to correct for damage in whole genomes and apply some of these high-resolution new methods on. That's how we can really understand finer-scale history in the future. And so I think understanding this will allow us to understand genetic history in finer scale, which is interesting to geneticists such as myself. But I think also understanding it on a per individual level will be very relevant to archaeologists and perhaps historians. And for that, we need high-quality ancient genomes and more of them as well. Thank you very much.